Properties of cardiac muscle is a bit complicated topic, but it is quite important for Viva, as well as it forms the basics of cardiac physiology. In this lecture, I am going to make you confident for this topic, and by the end of this session, you will be having the notes for the same. So let's start. Hey everyone, welcome to my channel. I am Harshita, the founder of HM Learnings, where students come to clear their doubts and to get their study material. Make sure that you have subscribed the channel. So let's start. Firstly, we will talk about the physiologic anatomy of cardiac muscle. That is the histology of cardiac muscle. So if you look at this structure, this figure, you can see that the cardiac muscle is cylindrical in shape. The next point is the cardiac muscle cell is uninucleated, means there will be one nucleus per cell. Not like the skeleton muscle where there is multiple nuclei per cell. Then it is involuntary, means we can't influence its functioning. Then it is striated. Striated means there will be striations on its surface because of the presence of thick and thin filaments. Lastly, it is branched. Not like the skeleton muscle, which shows the parallel arrangement of the myofibrils. So here the cardiac muscle, this is one cardiac myocyte. So this one cardiac myocyte, it branched at the ends. It will branch at the ends to fuse with the another cardiac myocytes. So this one cardiac cell will fuse with the another two cardiac myocytes, and it will form a characteristic 3D structure. In the skeleton muscle, it is not like that. Now these two cells, these two cardiac myocytes, they fuse at the membrane. They fuse at the membrane. Both the cells membrane will fuse, and they form a structure called as the intercalated disc. So this section, this dark color portion, is basically the intercalated disc, which is nothing but the fused cell membranes of the two cardiac myocytes. So the intercalated discs are basically the specialized junctional areas or the regions which usually occur at the Z line of the sarcomere. So sarcomere has a Z line. At the Z line, the intercalated disc is present. So looking at the intercalated disc has two types of the junctions. It has anchoring junctions and it has electrical junctions. Anchoring junctions are beyond the scope of this video. I will make a separate video on the anchoring junctions, but in this video we will limit our discussion on the electrical junctions only. That is the gap junctions. So we have seen the diagram of the cardiac myocyte. I am drawing here again. So this is one cardiac myocyte cell which branched out at its end, and it will fuse with the another two cardiac cell, and these are the nucleus. So it fused at the intercalated disc. An intercalated disc is nothing but the fused cell membrane of the adjacent cardiac myocyte. So if I zoom out this region, so this is number 1 cardiac myocyte and this is number 2 cardiac myocyte. So you can see that the intercalated disc is a series of multiple folds. It is not a straight line. It is a series of multiple folds. Also, this is cardiac myocyte 1 and this is cardiac myocyte 2. So between two cells there is a space, and that space is called as the intercellular space because it is between the two cells. So this intercellular space is about 20 to 25 nanometer. Now look at the diagram here. This is not a cardiac cell diagram. This is a diagram of epithelial cell I have taken to make you understand the gap junctions. So this is epithelial cell 1 and this is epithelial cell 2. So between these two cells, there are many types of the intercellular junctions. We will not discuss other types of intercellular junctions. We will discuss only about the gap junctions. But this is just to show you a major difference between the other types of the junctions and the gap junction. So between these two cells there is intercellular space. So at the other sites, at the other intercellular junctions, the intercellular space is more. But when you look at the gap junction, this is the reason where the gap junctions are present, the intercellular space has been narrowed down. It has been decreased, and it is decreased up to what? To 3 to 3.5 nanometer. So the intercellular space is reduced by the gap junctions. From 20 to 25 nanometer, the intercellular space is being reduced to 3 to 3.5 nanometer. And this is the function of the gap junction. They will decrease the intercellular space between the two cells. So, what are the proteins which make the gap junctions? So, in the next slide, we will look at this point. So, this is a diagram I have taken from the Gannon Review of Medical Physiology. Here, this is the first cardiac myocyte. This is one cardiac cell, and this is the another cardiac cell. So, between these two cardiac cells, there is intercellular space of about 20 nanometer. And this space will be narrowed down to about 3.5 nanometer, where the gap junctions are present. So again, the gap junctions narrow down the intracellular space to 3.5 nanometer. Now the gap junctions are made up of the proteins called as the conexon. So conexon is a protein which makes the gap junction, and conexon has 6 subunits. It is basically what? It is a hexamer. So 6 subunits of connexin. So 6 connexins will come and they will make the connexon. So here this is written 6 connexin, that is subunits, will form the one connexon. So this is a diagram of a one connexon. If you will count here, there will be 6 subunits of connexin. 1, 2, 3, 4, 5 and then 6. One connexon will form hemichannel. Hemichannel means half channel. So another connexon present on the another cell will form the another half of the channel. So if I draw here the cell membrane of one cardiac cell having one connexon and the another cardiac cell having the another connexon, these two connexons will fuse and they will form a gap junction. So from this passage, you can see here, this forms a passage or the tunnel through which the ions or the other solutes can pass from one cell to another cell very easily. Okay, so from one cell to another cell they will pass very easily. So gap junctions reduce the intercellular resistance. The intracellular resistance will be reduced. The ions can be passed very easily from one cell to another cell. So because of the help of the gap junctions, the heart can act as a syncytium. So what is syncytium? We have two types of syncytium. One is the anatomical syncytium. So the anatomical definition of syncytium is that you have a cell and that cell has multiple nuclei. That cell has no division. Okay, this is one cell which has many nuclei, multinucleated cell, and it will function as a single cell because there is no separation between these nuclei. But when you talk about the functional syncytium, what is happening here? There are many cells. There are many cells and each cell is Each cell is divided from the another cell by the presence of a cell membrane, and each cell will contain one nuclei. But it is acting as a functional syncytium. Even though there is a plasma membrane, there is a boundary between the two cells, still it is acting as a single unit. And this is possible because the passage of the ions between these cells is very rapid and very convenient because of the presence of gap junctions. And that is why the heart, even though the cardiac muscle is uninucleated, it has cell membrane, but it will act as functional syncytium because it has gap junctions. So there are two syncytiums in the heart, the atrial syncytium and the ventricular syncytium. Atrial syncytium means the whole atrium will act as a single unit. It will contract at a same time. As well as the ventricles, they will also act as a single unit and will contract at the same time. These two syncytiums are separated by what? AV, that is atrioventricular fibrous ring. And that fibrous ring is not conducting the impulse directly from the atrium to the ventricle. And the impulse is basically carried by the AV node from the atrium to ventricle. So there is one atrial syncytium which will contract together, and there is another ventricular syncytium which will contract together. Okay, so by this you can understand the significance of gap junctions in the cardiac muscle. So we look at the properties of cardiac muscle. It is divided into the electrical properties and the mechanical properties. We will look at each and every property of the cardiac muscle one by one. So first is the excitability. Excitability is defined as the ability of the cardiac muscle to respond to different stimuli. So like every nerve and every muscle, the cardiac muscle is also excitable. It will respond to the electrical stimulus. It will respond to the any kind of stimulus which is coming from the external environment, as well as if it is originated or generated by its own. The resting membrane potential of a ventricular cell is minus 90 millivolts, but that of the SA node is minus 70 millivolt. So why there is difference, and what are the two types of action potential? What are the two types of the electrical activity in the heart? The fast response action potential and the slow response action potential we will cover when we discuss about the electrophysiology of heart. Till then, you just remember that the cardiac muscle is excitable, and it is the ability of the cardiac muscle to respond to different stimuli which is called as the excitability. The excitability is also called as the bathmotropism. Moving to the next property, which is the automaticity. So automaticity means what? The thing is automatic. It occurs by its own. It doesn't need any external stimulus. The heart doesn't need any external stimulus to beat. And this is because of the presence of the specialized cardiac muscle cell which have the ability to generate its impulse at a regular rate and in a regular rhythm. And this is basically done by the SA node, which is also called as a primary pacemaker of heart. So SA node has a different type of the potential called as a pacemaker potential, through which it can depolarize by its own. It doesn't need any external signal to go to threshold and then to depolarize. It will automatically depolarize itself. And hence, the normal heart rate is under the control of SA node. The normal discharge rate of SA node is between 60 to 100 beats per minute. As a result, the normal heart rate will be under the range of 60 to 100 beats per minute. Now the next property is conductivity. So the impulse is generated in the SA node because SA node is the primary pacemaker of heart. Now it has to go to every part of the heart. Each and every part of the heart has to receive the external stimuli. Then only the cardiac muscle will start contracting. So this function will be done by the conducting system. This function will be done by the cells which will conduct the impulse at a faster rate. And this is called as the dromotropism. Okay, so the next property is contractility. So if the muscle receives an electrical stimulus, what it will do? It will give a mechanical response. It will contract in response to that. So the cardiac muscle contracts in response to the electrical stimulus, and this is also called as the inotropism. So now we know that the heart is not under the control of brain. It doesn't need stimulus from the brain, but yet the brain can influence its activity via the help of autonomic nervous system. So autonomic nervous system has two divisions. It has parasympathetic nervous system and it has sympathetic nervous system. The parasympathetic nervous system will do everything in negative in the case of heart. It will decrease the excitability, that is negative bathmotropism. It will decrease the heart rate, that is negative chronotropism. It will decrease the contractility of the heart, that is negative inotropism, and conductivity will also be decreased by the parasympathetic system, that is the negative dromotropism. Looking at the sympathetic nervous system, all these things are positive. There will be positive bathmotropism, positive excitability, there will be positive chronotropism, positive inotropism, and positive dromotropism. So here are two very important statements which you need to remember. That heart rate in a normal situation is predominantly under the influence of parasympathetic nervous system. If the heart is denervated, means if the innervation is cut off, then in that case what will happen? The resting heart rate will be more than 100. This is because in the normal heart, when the heart has innervation, the parasympathetic nervous system lowers the heart rate, and that's why it falls under the range of 60 to 100 beats per minute. Then if you look at the another thing is that the negative inotropism is not that much shown by the parasympathetic nervous system. Okay, so these are the two statements you have to remember. We can say that it shows a negative inotropism, but not that much. The next property is refractory period. Cardiac muscle have a long refractory period. Now the question arises, what is refractory period? The refractory period is the period during which the muscle will not evoke any response to the stimulus. So the stimulus has came, but the muscle is in refractory period. So the muscle will not respond to that electrical stimulus. There are two types of refractory period, the absolute refractory period and the relative refractory period. Absolute refractory period is that period during which the stimulus of whatever strength, of whatever potential, of whatever intensity is not able to evoke any response from the muscle. It will be followed by the relative refractory period, during which the suprathreshold stimulus can evoke a response. Now, what is the role of a long refractory period? Long refractory period means that the muscle will be not able to respond to the second stimulus for a longer duration because it is having a long refractory period. So what is the role or the importance of this long refractory period? The duration of action potential in cardiac muscle is about 250 milliseconds. Now look at the two graphs. These two graphs, I label it first. This is a graph of cardiac muscle and this is the graph of a skeleton muscle. So if you look at the first part of this graph, that is this graph, this is a graph of action potential. Now this is a graph of action potential, and we know that in the action potential we have what? We have action potential has depolarization phase and it has repolarization phase. It has two phases. So if you look at this graph, you can see that this is depolarization and the remaining one is the repolarization. So the depolarization is very rapid. It is lasts for about 2 milliseconds, and rest of the part of the 250 milliseconds will be taken up by the repolarization. So the repolarization is very, very long, and it is long because of the plateau phase and the slow repolarization which is happening. So this repolarization has the absolute refractory period and the relative refractory period. Now when we look at the skeleton muscle, this is a diagram of a skeleton muscle. So again, this is a graph of a action potential. So this graph of action potential is very different from this graph of the action potential. Here there will be rapid depolarization which will be followed by the rapid repolarization. So here there is no elongation of the repolarization phase, there is no plateau phase. So the action potential duration in the case of skeleton muscle is hardly 2 to 5 milliseconds. Okay, and if you look at the duration of the absolute refractory period, it is also very less, about you can say about 4 milliseconds, and the remaining 1 millisecond or about 0.5 milliseconds, it is just a rough idea, is a relative refractory period. But when you look at the duration of the absolute refractory period in the cardiac muscle, it is ranging from for the first 200 milliseconds, and the remaining 50 milliseconds is the absolute refractory period. So the refractory period is very, very long in the case of cardiac muscle, and it can be easily understood by these two graphs. Now we look at the muscle twitch graph. So what is muscle twitch? Muscle twitch is basically a graphical recording of the mechanical response of the muscle to the electrical stimulus. So this is the muscle twitch of cardiac muscle, and this is a muscle twitch of a skeleton muscle. So muscle twitch has latent period, then a contraction phase, and then a relaxation phase. So here is a latent period. So latent period is what? A period, you can say the time gap when the muscle receives a stimulus and the beginning of the muscle to respond that stimulus. It is a time gap between when the muscle receives a stimulus and when the muscle begins to respond to that stimulus. So here the latent period in the cardiac muscle is very, very less, but in the skeleton muscle the latent period is very large. Okay, it is much more than the cardiac muscle. Then there is a contraction phase, and then there is a relaxation phase. So the duration of muscle twitch in the cardiac muscle is 1.5 times of the action potential duration, and it is about 375 milliseconds. And the muscle twitch duration in the skeleton muscle, it is 100 milliseconds. So you can see the difference. The difference is that much of the mechanical response of a cardiac muscle is finished when the muscle is in action potential. But here what is happening in the skeleton muscle? The muscle twitch is much longer than the duration of the action potential. Means that the action potential overs, but the twitch, the mechanical response of the muscle is going. Okay, so now we can say a very important point here, that because of this difference, what is happening, the skeleton muscle can be tetanized, but the cardiac muscle can't be tetanized. So here you can look that this is the contraction phase. So the contraction phase is over much before the repolarization is over. This is repolarization. The repolarization is ongoing, but the contraction is over much before the end of the repolarization. But here what is happening? The absolute refractory period is over, and the relative refractory period is also almost over when the contractile response is reaching its peak. And then there is relaxation phase. So that is why the cardiac muscle can be tetanized. So what is tetanization? Tetanization means that suppose a second stimulus of a suprathreshold intensity has came when the muscle is in relative refractory period. But when we look, this is the first stimulus of the muscle. This is the first action potential of the muscle, and this is the first contractile response of the muscle towards that action potential. So the muscle, when it is in the relative refractory period, it will be starting its contractile response to the first action potential. But the second stimulus has came in the relative refractory period of the first stimulus, and the muscle is in contractional phase of the first stimulus. But because it is of suprathreshold intensity, the muscle will give its contractile response, and the strength of the contraction will be increased. So this is a strength of the contraction which is happening in the first phase, but because the second stimulus has came in the relative refractory period and the mechanical response is ongoing, the muscle is starting its contraction, the strength of the contraction because of the second stimulus will also be happening. There will be more calcium entering, and hence the strength of the contraction will also be increased. And this will happen if the third stimulus also came in the relative refractory period of second stimulus, and similarly it will happen. This is called as a tetanization. So in the tetanization you can see there is hardly any relaxation. There is no relaxation at all. And this can't be possible in the cardiac muscle, because the contraction phase is over in the absolute refractory period, it is over in the repolarization. So if the second stimulus came, till then the muscle will complete its relaxation also. So what is the importance that the cardiac muscle can't be tetanized? It should not be tetanized. If it is tetanized, what will happen? There will be no relaxation. The cardiac muscle will not be able to relax. And when the cardiac muscle will relax, there will be filling of the blood in the ventricles. So if there is no relaxation, no filling. No fillings will be what? There will be no cardiac output. Okay, so it will be a very fatal condition. So I hope that you get the concept. These are the very important questions repeatedly asked in the Viva as well as in 1 and 2 marks questions in your university examination. Why the cardiac muscle is not tetanized and why it should not be tetanized. So I have written the answers, you can note it down. Then we move to the next property, that is distensibility. Distensibility means the ability of the muscle to stretch. The ventricles, they have a property of distensibility so that they can accommodate the blood during diastole without much increase in pressure. Each time when a blood came, the ventricle has to relax, it has to stretch, and it has to accommodate blood without increase in its pressure. And it is a very, very important property of the heart. Moving to the next property, that is a length-tension relationship, which explains the very important law of the cardiac physiology, Frank-Starling law. So according to Frank-Starling law, the force of contraction is directly proportional to the initial length of the muscle fiber within the physiological limit. So what does it mean? So if you look at this graph, you can see that on the y-axis we are plotting a muscle tension, and on the x-axis we are plotting a muscle length. So What is passive tension or what is resting tension? The passive tension and the resting tension, they are the synonyms. So what is passive tension? Passive tension means that when the muscle is in relaxed position and we are trying to stretch it. So we are applying a force to stretch it, and that tension, that force required to stretch the muscle when the muscle is at rest is the resting tension or the passive tension. So this resting tension or the passive tension is because of the presence of the titin and the other elastic elements in the muscle. So what is the passive tension in the case of heart? In the case of cardiac muscle, what is the passive tension or what exerts the passive tension on the cardiac muscle? So it is the end diastolic volume or the preload. Now suppose this is left ventricle. Now left ventricle will receive the blood during the diastole. So this blood received during the diastole is called as a preload. So if more blood came, what will happen? There will be more stretching of the left ventricle. There will be more stretching of the cardiac muscle. Okay, and that means there will be more passive tension, more resting tension on the muscle. So this graph shows that increase in the preload, increase in the tension, increase in the force will increase the resting tension on the muscle. Now take an example of rubber band. You want to stretch the rubber band, you are applying a certain kind of force on it. You are stretching it. You are applying which force? Resting tension. So more you want to stretch, more you have to apply the resting tension. Similarly, more you want to stretch the cardiac muscle, more blood has to go in the diastole. More should be the preload in the diastole. So now the next is what? The active tension. Another term is active tension. So active tension is developed because of the formation of the cross bridge between the actin and the myosin. So now again take an example of rubber band. You are stretching it, you are applying a force. So more force, if you are applying more force, what will happen? The rubber band has more tendency to again come to its original shape and size. Okay, similarly on the cardiac muscle, the more you stretch, what will happen? Means the more the preload, the more the resting tension, more will be the force of the contraction of the cardiac muscle which will be generated. The cardiac muscle will contract more in response to the more preload, because more resting tension means there will be more force of the contraction. Just like the rubber band, more force you applied, more forcefully it will come back to its original position. So this is what said by the Frank-Starling law. The force of contraction is directly proportional to the initial length of the muscle fiber, and the initial length of the muscle fiber is depends upon what? The preload. Okay. Then what are the mechanisms which explain the Frank-Starling law? So there are three mechanisms which are proposed to explain this mechanism. Okay, why there is increase in the force of the contraction when there is increase in the preload. So number one is the cross bridge formation. Cross bridge formation between actin and myosin generates which tension? Active tension. Okay, more active tension means more force of contraction. So peak tension will be developed, means maximum tension will be developed when the sarcomere length is about 2.2 to 2.3 micrometer. Why? Because this is the optimal length. Optimal length means there will be maximum number of cross bridge formation which occurs here between the actin and the myosin. So this is a length at which there will be maximum overlapping of the actin and the myosin. Hence there will be maximum number of the cross bridge will be formed, maximum active tension will be generated, and maximum force of contraction will be generated. What will happen if there is more overlapping? If there is decrease in the length of the sarcomere less than this, so at that time what will happen? This is a structure of a sarcomere. Okay, these are the thin filament, this is the thick filament. Okay, so what happens when the tension, when the sarcomere length is less than 2 micrometer? So in that case, what will happen? The opposing thin filaments will be starting overlapping each other. So there will be no that much formation of the cross bridges, and hence the tension developed will be decreased. If the sarcomere length is increased, what will happen? There will be again no significant overlapping between the actin and the myosin. So again, number of cross bridges formed will be decreased, and hence what will happen? The force of the contraction will be decreased. Second mechanism is length dependent activation. More length, more there will be sensitivity of the troponin C to calcium, and hence there will be increase in the force of contraction. More stretch, more opening of the stretch sensitive calcium channels. More calcium channels, more entry of calcium, more intracellular calcium concentration, and more force of contraction of the heart. So these three mechanisms explains the basics of the Frank-Starling law. The next property is load velocity relationship. So here on the y-axis, velocity of shortening of the muscle, and on the x-axis, load on the muscle. So load on the muscle is called as the afterload, means the resistance in the vessels against which the heart has to pump. So if there is more resistance against which heart has to pump, there will be decrease in the velocity of shortening. If there is more load on you, what will happen? Your excitement or your energy will be lowered down. Similarly for the heart, if there is more load on muscle, the shortening of the muscle will be decreased, and this is called as a load velocity relationship. Another property is the force frequency relationship. The force frequency relationship explains the staircase phenomenon, also called as Trippe effect or the Bowditch effect. So what is happening in this effect? When there is increase in the heart rate, means when there is increase in the frequency of the stimulation, or you can say the discharge rate of the SA node, when there is increase in the heart rate, there will be increase in the force of the contraction of the cardiac muscle. So as the discharge rate of the SA node is increased, as there is positive chronotropism, the force of the contraction of the heart will be increased. The cardiac output will be increased. So in this graph, you can see what is happening. The force of the contraction, the strength of the contraction is continuously increasing. So why it is increasing? Because the stimulus has came when the muscle is in the relaxation phase. The relaxation phase is not completely over, but another stimulus has came which causes the another generation of the mechanical response, and here the strength of the contraction will be increased, will be more than the previous stimulus. So what are the mechanisms which explains the staircase phenomenon or the Trippe effect? So there are three mechanisms which explain the staircase effect. Number one is the increase in the sarcoplasmic calcium concentration. So before that, first you understand the basics behind this process. So what is happening, that when there is increase in the heart rate, so there is increase in heart rate, means there will be increase in what? There will be increase in action potential frequency. If the frequency is increased, what will happen? If the frequency is increased, that means the duration has to be reduced. Why? Because the number of the action potential, the number of the times the SA node is discharging is increased. So definitely what will be decreased? The duration will be decreased. So when there is increase in heart rate, there is increase in action potential frequency but decrease in action potential duration. So if there is increase in action potential, decrease in action potential duration, there will be decrease in depolarization phase, there will be decrease in repolarization phase. The refractory period will also be reduced. If there is decrease in depolarization, the systole duration will also be decreased and the diastole duration will also be decreased. Here, if there is decrease in action potential frequency, there will be more number of depolarization, more number of repolarization. The frequency is more, but the duration is decreased. Okay, so here what is happening? Now you can easily understand the mechanism. So as there is increase in the number of the times the cell is depolarizing, what will happen? The number of the times the calcium is entering inside the cell will also be increased. But the relaxation phase, what happens when there is relaxation? When there is relaxation, the calcium has to exit. It has to came out of the sarcomere. It has to came out of the cardiac cell. But the rate of the entry of the calcium is more than the rate of the calcium exit. Means number of calcium which is entering is actually faster than the rate of the calcium which is going outside the cell. As a result, there will be increase in the sarcoplasmic calcium concentration. So why this is happening? This is because the sodium potassium ATPase pump is not able to maintain the electrogenic gradient which is required by the sodium calcium exchanger. The working of the sodium calcium exchanger depends upon the working of the sodium potassium ATPase. The sodium potassium ATPase has to maintain the sodium concentration outside the cell higher and inside the potassium concentration higher. So it will pump 3 potassium outside the cell and it will pump 3 sodium outside the cell and it will take 2 potassium inside the cell. So what is happening? Ultimately, the intracellular concentration of sodium will be reduced. So what is happening? There will be establishment of the concentration gradient for the sodium because sodium is lower inside the cell. So concentration gradient will be established from outer to inner. So sodium will start moving inside the cell along its concentration gradient, and in exchange to that there will be calcium which will move outside the cell. So because there is decrease in the activity of the sodium potassium ATPase pump with the increase in the heart rate, there will be accumulation of the calcium because now the sodium calcium exchanger is not able to maintain the lower sarcoplasmic calcium concentration. So just remember that the rate of the calcium entry is more than the rate of the calcium exit. As a result, there will be sarcoplasmic calcium concentration. More calcium means more will be the force of the contraction of the heart. The second mechanism is after first contraction the temperature will increase and this temperature will favor the enzymatic activity for the muscle contraction. After some contraction, increased temperature will decrease the viscosity of the sarcoplasm. So the sliding of the filaments, that is the sliding of the actin over the myosin, will be easier. So these are the 3 mechanisms which are repeatedly asked in the vivas by the examiner, that what is staircase phenomena and why it is happening. What is the mechanism? These are the references you can look forward for this topic. This topic is also available in the Hindi version. I will share the link in the end screen as well as in I button. You can also find the link in the description box. Go and check the video there if you find Hindi language more comfortable. Subscribe to HM Learnings, like, share and comment. Let me know if you have any doubt. Till then, keep learning.